Lesson 2.4, Scientific Method. Our guiding questions and objectives. What are the steps of the scientific method? How to write a hypothesis for experiments, identify variables in an experiment, and use CER to write conclusions. What is scientific inquiry? Scientific inquiry is the process by which scientists seek to answer questions. Here we have a picture of a student, and there are different parts of scientific inquiry, but the main idea is that inquiry is how we answer questions. So if you have a question, you may inquire, and that's what inquire means, to ask a question. So scientific inquiry is how we find answers to questions. We may do experiments, we may do just observations. I have here, you can do labs, research, experiment, and observations are a few ways that scientists can do scientific inquiry. Part of it, scientific inquiry, we do that through the scientific method. This is a way that scientists learn about the world, and it's more organized, and it's good to keep it that way so that scientists can make predictions and then test those predictions to see if they are correct. So here are the steps. Make observations, ask questions, do research, make hypotheses, perform an experiment, collect data, analyze results, and write a conclusion. Now we're going to go through each of these steps. The first step is to make an observation. This is actually something that happened this summer. This is a zucchini plant that I had, and I found these little things underneath. So my observation is, what are these small egg-looking things under my zucchini leaves? I asked some questions. What are they? Are they harmful to the plant? The next step in the method is to do research. So I Googled it. What are bugs, like eggs underneath my zucchini plants? And I found pictures, and I said, yeah, I've seen those bugs on my plants. They are squash bugs, and they are harmful to the plant. So what do I do to get rid of them? That is, I'm using the scientific method to answer this, solve this problem. So you can form a hypothesis. A hypothesis is a prediction that can be tested. Your hypothesis should explain what you expect to happen, be clear and understandable, be testable, be measurable, and contain the independent and dependent variable. We'll talk about that in a little bit. Here is my hypothesis for an experiment that I can conduct about these bugs. If I spray my plant with peppermint oil water mix, then the bugs will leave me alone. It's important that you use an if-then statement for your hypothesis, and we can practice this more. But if I do something, then this will be the result. It's very clear what I'm going to do and what I expect to happen. I can set up an experiment to do this. If I spray the plant with peppermint oil and the bugs didn't leave it alone, then my hypothesis is wrong. But if the bugs do leave the plant alone, then maybe my hypothesis was correct. So this would be a great hypothesis that meets all of these things. Next, I can do an experiment. So here's an idea of an experiment. I have 3 squash plants. This one gets full sun. This one gets 2 hours of sun. This one gets no sun. This one is watered 2 times a day. This one's watered 2 times a day. This one's watered 3 times a day. I sprayed this first plant with peppermint oil. I spray this plant with peppermint oil. And I do not spray this plant with peppermint oil. Is this a good experiment? If you think yes, why? If you think no, why? This is not a good experiment. There are way too many variables being changed. If this one has no bugs, is it because it had peppermint oil? Or is it because it has a better amount of sunlight? Is this one too dry? The bugs don't want to live over there. There's not a clear cause and effect. And so let's look at what are variables. Variables are the parts of the experiment that can be changed. How much sun the plant gets, how much water the plant gets, if the plant gets sprayed with oil or not. Those are all variables. The manipulated variable is the one thing that we change on purpose. There should only be one thing that you're testing at a time. The responding variable is the results. So what happens as a result of that manipulated variable? And then constant variables, I'll move me over here. Move. Those are the things that we keep the same. And so it's important that we keep everything the same except for this. And so I'm going to look back here. Here, we didn't keep the amount of sun the same. We didn't water it the same amount. We didn't do this the same. So we had way too many manipulated variables. There was too many things that we changed that were different about each plant. So here's a good example. So what was our independent variable? It's also known as a manipulated. I'm sorry, but you'll see it interchangeably. So manipulated variable, independent variable. Our example here is the peppermint oil water spray. So that's the cause. That's what we're going to change. We want to know, how does that affect the number of squash bugs? But there's other things that are influencing our number of squash bugs. These are the constant variables. So how much rain or how much water they get. So we should water them the same. The breeding season, what time of the month are we doing our experiment, or what time of the year? We have to make sure that we treat all plants at the same time, because that is another constant, or that can be another variable. And then finally, how much sun are our plants getting? We want to keep all of these things the same so that they don't affect this. We want only thing that affects the number of bugs is the peppermint oil. So here's a better example. They all get full sun. They all get water 2 times a day. These are our constant variables. We are going to keep these constant. We're controlling those. Then the spray, this one doesn't get spray, this one does, this one does. Those are our manipulated variable, if they get the spray or don't get the spray. One more thing, there's 3 different groups here. There's 3 different treatments, or 3 different experimental groups. This one is called our control group because it is not getting the treatment. We need to have a control group so that we can compare our results to these. If they all still have the same number of bugs, we can say, well, peppermint oil didn't really do it. If all the bugs go away here and they're still there here, we know, okay, without the treatment, they would have stayed on there, but with the treatment, they're gone. So those are variables. Next, we need to collect and analyze data. This is done in a notebook, or you can do it on the computer. Make sure you have your units and the date that you collected it. Here would be what it could look like. So maybe I have my date here, the bugs on this plant, the bugs on this plant. You keep track of the numbers. You know that this number means how many bug eggs. So it's good to have the units. Then you form a conclusion, and this could be a whole video of itself. We'll do lots of practice. Your conclusion should be the end of the report or study. You should use complete sentences, and there are 3 main sections that we want to use: claim, evidence, and reasoning. And so you should have a sentence that is your claim, the answer to the question. Did the peppermint oil work? Your evidence. Give evidence from your data, specific numbers and facts that you collected that help support that claim. And then your reasoning would be, why did that oil work? So maybe in your research, you found that they didn't like the smell of the oil or something about that. You can include that in your reasoning. So there will be extra help for this later.